This is Learning to Grow, Control of Material Self-Assembly Using Evolutionary Reinforcement Learning. I'm Steve Whitelam of Lawrence Berkeley National Lab, and this is joint work with Isaac Tamblyn. Molecular self-assembly is a way of making materials, whether something simple like a colloidal crystal, or something more elaborate like the surface layer protein lattice that forms on the outsides of certain bacteria, or a metal organic framework used in gas capture. Despite many successes in the laboratory, we're yet to realize the full potential of self-assembly because it's a non-equilibrium process, and our understanding of non-equilibrium processes is much less complete than our understanding of equilibrium phenomena. So given a prediction for material, how do you make it? If we could answer this question, we could imagine the material we would like, and then work out the protocol we would need to make it. So as proof of principle, we can investigate this on the computer using highly simplified models. So it turns out that you can capture some of the complexity of different types of material, DNA, nanotechnology, organic molecules on surfaces, using highly simplified statistical mechanical models that account only for coarse features of the molecules, such as their symmetries and binding energy scales. And these simple models can reproduce many of the structures and phenomena seen in and out of equilibrium in a wide variety of molecular systems. So we can use this system as a test for learning protocols using the following environment. So we're modeling molecular deposition on a surface, and the control parameters we're interested in are chemical potential and temperature. And so in the picture shown on the left, the model molecules or disks live on a surface, and they can exchange with a notional bath with a certain chemical potential, and they're held at a particular temperature, which relates to how strongly they bind to each other. In usual protocol design, you might hold the temperature constant or change it slowly as a function of time. And in this work, we encoded the temperature and chemical potential as functions, as neural networks, and evolve neural networks in order to promote certain outcomes. The inputs to the network can be as simple as the elapsed time of the protocol, or sometimes some microscopic information that might be accessible experimentally. Networks, initially chosen at random, promote random protocols and low-quality assembly, but under neuroevolutionary learning, they learn to promote desired outcomes specified by particular order parameters. As an aside, I should say that we're using neuroevolution of the weights and biases of a network. In recent work, we demonstrated a formal equivalence between this kind of neuroevolution and gradient descent in the presence of Gaussian white noise. So this demonstration reveals the particular connection between two ways of training neural networks and provides some justification for the empirical success of neuroevolution. So these protocols are very effective compared to the human design protocols we already knew about. So in this example, we're trying to promote the self-assembly of a network made up of pores with 12 sides. So in the lower plot, the order parameter is the number of 12-sided pores formed in the simulation box as a function of time on the horizontal. So the neural network learned protocol is shown at the left, and it's much more rapid and much better than any of the slow cooling protocols shown on the right-hand side of the plot. So the three rightmost trajectory swarms are cooling quickly, moderately slowly, and very, very slowly. And these take days or weeks of computer time. So despite the simplicity of the model, the assembly of this particular structure is non-trivial, and evolutionary learning produces protocols that are better than those we knew about previously. They can also be used for polymorph selection. So polymorphs are different physical structures of the same set of molecules. Polymorph selection is important, for instance, in drug synthesis, where one polymorph might be a valuable drug and the other a deadly toxin. Here we're considering two different types of polymorph, one with 6-sided pores, the other with rhombic pores. These structures built from the same set of molecules are isoenergetic. They have the same energies and very simple, similar nucleation-free energy barriers, so that simple, slow cooling produces either or both of these polymorphs in an uncontrolled way. So in the bottom plot, we're showing the two order parameters describing the two polymorphs on the two axes, and the circles result from 50 independent simulations in which we cool slowly. Under evolutionary learning, we managed to learn protocols that produce either one or the other of these polymorphs in a reliable way. So the colored symbols represent 50 independent simulations using the best protocol learned upon specifying one or the other order parameters as the genetic search parameter. So evolutionary learning can be applied to a wide range of synthesis, manufacturing, and self-assembly processes. And we're currently using it to study vapor deposition of glasses. And in addition, we're interested in the theoretical underpinnings of the kinds of learning used here.